Hello and welcome to some biology class. Friends, today we shall discuss about stomata. Stomata are the microscopic pores present in the epidermis of leaves, stem and other organs of the plant. They control the rate of gas exchange between the plant and the atmosphere. They are most abundant on the leaves. The singular form of stomata is stoma. This figure shows the structure of a stoma. A stoma has a central pore known as stomatal aperture. This pore is the stomatal aperture. This aperture is bordered by a pair of specialized parenchyma cells known as guard cells. These are the two guard cells that border the stomatal aperture. The guard cells are responsible for regulating the size of the stomatal opening. The guard cells are surrounded by a variable number of epidermal cells known as subsidiary cells or accessory cells. The subsidiary cells may be morphologically similar or different from the surrounding epidermal cells. The stomatal aperture, the guard cells and the adjoining subsidiary cells together constitute the stomatal apparatus. The guard cells are the specialized parenchyma cells that regulate the stomatal opening. In dicot plants and in non-grass monocotyledons, the guard cells are kidney-shaped. These are the kidney-shaped guard cells. Whereas in grass, the guard cells are dumbbell-shaped with bulbous ends. These are the bulbous ends. And in such type of stoma, the stomatal aperture is a long slit that is located between the two handles of the dumbbells. In such type of stoma, the guard cells are always flanked by two subsidiary cells. In dicot plants, more number of stomata are present on the lower surface of the leaf than the upper surface. Whereas in monocotyledons, stomata are equally distributed in the upper surface and the lower surface. In amphistomatic leaf, stomata are present on both the upper and the lower surface. And in hypostomatic leaf, stomata are restricted to the lower surface. And in epistomatic leaf, stomata are mainly present on the upper surface of the leaf. In aquatic plants with floating leaves, stomata are confined to the upper surface. And in submerged leaves, stomata may be altogether absent. The position of the stomata with respect to the epidermal cells varies. In case of mesophytes, the stomata is present at the same level as that of the epidermal cells. In case of xerophytes, which grow in dry habitat, stomata are located in a cup-shaped depression. Such type of stomata are called as sunken stomata. The main purpose is to reduce the water loss. Whereas in case of some other plants, stomata are slightly raised above the surface of the epidermis. The thickening of the wall of the guard cells play an important role during stomatal movement. The wall of the guard cell is differentially thickened. Due to this differential thickening, some portions of their wall are substantially thickened. This differential thickening results in a much thick outer wall and inner wall, a thin dorsal wall and a somewhat thickened pore wall. The alignment of cellulose microfibrils in the wall of the guard cells play a regulatory role during stomatal movement. In kidney-shaped guard cells, The cellulose microfibrils fan out radially. Due to this radial arrangement of the cellulose microfibrils, the guard cells curve outward during stomatal opening. The guard cells are specialized cells which can sense various environmental factors like light intensity, quality, temperature, humidity and intracellular concentration of carbon dioxide. In response to various environmental factors, the guard cells give appropriate response. Stomata have been classified in various ways by various workers. According to the classification of Metcalfe and Chuck, there are 4 types of stomata. They are anomocytic type, anisocytic type, parasitic type and diacytic type. In anomocytic type of stomata, the guard cells are surrounded by a limited number of subsidiary cells which are indistinguishable from the other epidermal cells. That means the subsidiary cells are similar with the other epidermal cell. Whereas in anisocytic type, the guard cells are accompanied by 3 subsidiary cells. Here there are 3 subsidiary cells, out of which one subsidiary cell is distinctively small. In parasitic type of stomata, the guard cells are surrounded by one or more subsidiary cells, but the subsidiary cells lie parallel to the long axis of the pore and the guard cells. In diacytic type of stomata, the guard cells are surrounded by 2 subsidiary cells which are arranged at right angle to the long axis of the guard cells and the pore. In addition to these 4 types of stomata, 2 additional types have been recognized by Stes. One is actinocytic. In actinocytic type of stomata, the subsidiary cells are radially elongated, whereas in cyclocytic type, the subsidiary cells are arranged around the guard cell in the form of closed rings. This is about different types of stomata. Functions of stomata. Stomata have 2 main functions. First function is they control the rate of gas exchange between the plant and the atmosphere. Carbon dioxide, which is used in photosynthesis, diffuse into the leaf, and oxygen, which is released during photosynthesis, diffuse out into the atmosphere through the stomata. Similarly, stomata allow inward diffusion of air containing oxygen. Another function of stomata is transpiration. Transpiration is a process by which water is lost from the aerial parts of the plant in the form of water vapor. Through transpiration, excess water is lost into the atmosphere. By doing so, stomata help in regulating the water level of the plant. Stomata also reduce the water loss by its closing. So friends, this is about the structure and function of stomata. For more updates, please subscribe my YouTube channel Sam Biology Class. Please share your comments which are very valuable to me. Thank you very much.